Welcome to the deep dive. And usually when we start one of these, our source material is just massive.
Oh, yeah, definitely.
Right, like we're usually pulling from these dense multi-volume historical biographies or sweeping economic analyses. But today, the parameters of our mission are entirely different. We are taking a microscopic look at something incredibly brief.
Which is actually a really fun challenge.
It is, because our sole source text for this entire exploration is just a single Wikipedia stub. It's an entry about a tiny, relatively obscure organism. And our goal today, the challenge we've set for ourselves and for you listening right now, is to extract every possible insight, implication, and mystery from this one specific encyclopedic entry.
Yeah, we want to show how even the briefest scientific summaries contain these vast networks of biological complexity, not to mention historical context and the sometimes rigid, sometimes chaotic architecture of human knowledge.
Exactly. So, okay, let's unpack this. The star of today's deep dive is Ingenia. Ingenia, right? Ingenia. It's a genus of marine nematodes, which are roundworms. And the source material immediately hits us with a really striking statistical anomaly. I mean, the oceans cover, what, over 70% of the Earth's surface?
Yeah.
Hosting this incalculable diversity of marine life. Yet this particular genus, Ingenia, contains only one single known species on record. Just one. Ingenia mirabilis.
Yeah, and the term for that in biology is a monotypic genus. It's really quite unusual.
Because usually the genus has tons of species, right?
Exactly. In taxonomy, we are totally accustomed to seeing a genus function as an umbrella. Underneath it, you might have dozens or even hundreds of closely related species that have radiated out into various ecological niches.
So finding a monotypic genus in a group as unbelievably diverse as roundworms, it just immediately prompts a lot of questions.
It really does. You have to consider whether Ingenia mirabilis is this highly specialized relict species.
Like the sole survivor of an ancient family tree.
Right, the last surviving branch of a much older evolutionary lineage. Or, and this is perhaps more likely, if we are simply looking at an artifact of sampling bias.
Meaning we just haven't looked hard enough.
Precisely. The marine meiobenthos, that's the ecosystem of microscopic organisms living between grains of sand, is notoriously undersampled. So it is highly probable that Ingenia actually has a robust family tree. The other branches are probably just sitting in unexplored sediment on some coastline that no one has bothered to survey yet.
That makes a lot of sense, and we will definitely get into the specific biology and the coastal habitat of this nematode in a minute. But there is a disclaimer at the very top of this Wikipedia stub that we absolutely have to address first.
Oh, the disambiguation note. It is wild.
It's so wild. It's a standard formatting note, but the content is just bizarre. It reads, and I quote, "This article is about the nematode worm. For the dinosaur formerly known as Ingenia yanshini, see Heyuania."
Ingenia used to be the name of a dinosaur.
A dinosaur. How does a microscopic marine worm end up in a naming dispute with a literal dinosaur? And more importantly, how does the worm win?
Well, the resolution of that dispute comes down to something called the International Code of Zoological Nomenclature, the ICZN.
The ultimate rulebook for naming animals.
Exactly. It's the overarching framework that governs the scientific naming of all animals. And one of the foundational principles of the ICZN is the principle of homonymy.
Homonymy. So same names.
Right. It states that no two available names for taxa within the family group, genus group, or species group can be identical. If you are operating anywhere within the kingdom Animalia, the genus name has to be entirely unique across the whole kingdom.
So you can't have a genus of bird and a genus of insect sharing the same name.
No, because it would completely undermine the stability and universality of scientific communication. Think about it. When a modern researcher searches a global database for the term Ingenia, the system simply cannot return both a marine nematode and an oviraptorid dinosaur. It would be chaos.
Okay, but the dinosaur in question, the one that's now called Heyuania, was originally named Ingenia yanshini. Knowing that these rigid rules of the ICZN exist, how does a collision like this slip through the cracks in the first place? I mean, paleontologists and marine biologists are obviously operating in completely different spheres, but you'd think there would be a centralized registry to prevent this right out of the gate.
You would think so, but historically, that centralized registry existed only in print.
Ah.
Yeah, scattered across thousands of physical journals in dozens of different languages all over the world. The naming of the nematode Ingenia mirabilis was published in 1957. The dinosaur, Ingenia yanshini, was named decades later in 1981.
Okay, so a 24-year gap.
Right. And the paleontologist who named the dinosaur named it after the Ingenia Depression in Mongolia, which is where the fossils were actually found. Now, in 1981, there was no comprehensive, digitized, searchable global database of every single animal genus ever published.
It was just card catalogs and paper journals.
Exactly. A paleontologist working in the Soviet Union or Mongolia in the early 80s would have had absolutely no practical way of knowing that a marine biologist sitting in Germany had already claimed the exact same name for a microscopic worm 24 years earlier.
So they just published it completely unaware. But when the collision was eventually discovered, which I'm assuming happened as all these biological databases were being digitized and merged into the internet,
Yes, that's exactly when these conflicts started popping up everywhere.
the rules had to be enforced. And the really fascinating thing here is that the determining factor isn't which animal is larger or more famous or has more museum exhibits.
No, the determining factor is simply time. The ICZN operates on the principle priority.
First come, first served.
Literally. Article 23 of the Code explicitly states that the valid name of a taxon is the oldest available name applied to it. Because the marine worm was published in 1957, it held absolute priority. The name applied to the dinosaur in 1981 became what taxonomists call a junior homonym.
A junior homonym.
Right. And junior homonyms are invalid. They must be replaced. So the dinosaur had to be stripped of the name Ingenia and eventually absorbed the replacement name Yanshini.
It's just so funny to me. The entire situation highlights this blind, bureaucratic impartiality of taxonomic rules. You have a microscopic organism living invisibly in the sand, and it forced a massive paleontological rebranding simply because its paperwork was filed first.
It's a great victory for the invertebrates, honestly.
Truly. And that bureaucratic machinery is fascinating, especially when you consider the sheer scale of the databases running these checks today. But let's actually move from the naming bureaucracy to the physical environment of this organism.
Let's do it.
The source text gives us a specific geographic footprint. Ingenia is native to Brazil. So identifying a microscopic, free-living organism in a specific region of the Brazilian coast, that implies a very specific set of environmental adaptations, right?
Oh, absolutely. The Brazilian coast is subjected to a incredibly complex interplay of hydrodynamics. You have the warm, southward-flowing Brazil current interacting with varied local topography. That creates distinct zones of wave action, tidal flushing, and sediment sorting.
So when we talk about a marine nematode being native to this coastal sand, we aren't just saying it lives in the water. We are talking about an organism adapted to the interstitial environment.
Yes, the meiobenthos. The interstitial space is basically this three-dimensional network of microscopic water-filled gaps between individual sand grains. To a nematode measuring perhaps a millimeter or less in length, a sandy beach is a dynamic, constantly shifting labyrinth.
And adapting to that environment means surviving some serious physical trauma, right? The crushing force of shifting sand, plus constant chemical changes in the water.
The physical adaptations alone are remarkable. Nematodes in these environments often possess highly specialized cuticles.
And the cuticle being the outer skin.
Basically, yes. It's the tough, flexible outer covering of their bodies. And it has to be able to withstand the abrasion of quartz and silicate grains constantly grinding against each other under the wave action.
Like living inside a rock tumbler.
Pretty much. And they also have to navigate extreme chemical gradients. The interstitial water near the surface of the sand is well oxygenated by the waves crashing. But if you go just a few centimeters deeper into the sediment, the oxygen levels plummet. You get these hypoxic or even anoxic zones that are dominated by hydrogen sulfide.
So an organism native to this specific coastal environment has to be perfectly calibrated. It has to migrate up and down through these microlayers, seeking out optimal oxygen concentrations while also avoiding being washed out to sea by the tide.
Exactly. It's an incredibly demanding place to live.
The text gives us the foundational citation for how we even know about this specific organism's presence in Brazil. It points to reference number 1, a paper authored by S.A. Gerlach, published in 1957.
Yes, Sebastian Gerlach, a huge name in the field.
And the title of the paper is in German. Let's look at this. It's Die Nematodenfauna des Sandstandes an der Küste von Mittelbrasilien.
Excellent pronunciation.
Thanks, I try. And we can actually decipher the core meaning just by looking at the cognates, right? The Nematodenfauna is obviously the nematode fauna. The Sandstrandes is of the sandy beach.
Yep.
And der Küste is on the coast. And von Mittelbrasilien is central Brazil. But there's a parenthetical at the end of the title that really stands out to me. It says, Brasilianisch Meers Nematoden IV. or Brazilian marine nematodes, Part 4.
Part 4.
Yeah. What does the existence of a Part 4 tell us about the methodology and the sheer scope of the science being done here?
Well, the notation of Part 4 reveals that this single species description wasn't just a random one-off discovery. It was part of a massive systematic taxonomic campaign. In the 1950s, the whole field of marine biology was undergoing a significant post-war expansion.
Right, getting back out into the field.
Exactly. Researchers were launching these extensive sampling expeditions to catalog the biodiversity of the neotropics. These were regions that had previously been heavily undersampled compared to European coastlines.
So Sebastian Gerlach was out there doing the heavy lifting.
He was a towering figure in nematology. But conducting this work in 1957 meant relying entirely on painstaking, labor-intensive morphological analysis.
We should really detail what that analysis actually looked like in the 1950s because the publication venue is listed as the Mitteilungen aus dem Zoologischen Museum in Berlin, a zoological museum publication in Germany.
Right.
So a scientist is taking physical samples of wet sand from the coast of central Brazil, transporting those buckets of sand across the Atlantic Ocean to a laboratory in Berlin, and then having to somehow isolate and identify these invisible organisms without any modern digital imaging or genetic sequencing.
The extraction process itself is a highly specialized skill. First, you have to separate the microscopic meiofauna from the heavy sand grains. In the 1950s, this often involved decantation techniques.
Meaning washing the sand.
Kind of. You suspend the sand in water, vigorously stir it, let the heavy silica settle to the bottom for a few seconds, and then quickly pour off the suspended microscopic life into an incredibly fine sieve.
That sounds tedious.
Extremely. And once the nematodes were finally isolated, they had to be fixed, often in formalin, and then slowly transferred to anhydrous glycerin over several days.
Why glycerin?
To clear their internal tissues, it makes them more transparent so they can actually be viewed properly under a light microscope.
And then the actual identification relies on incredibly subtle visual differences.
Gerlach would have used a camera lucida.
What is that?
It's an optical device. It superimposes the microscopic image you're looking at onto a piece of drawing paper sitting next to the microscope, so he could trace the microscopic anatomy by hand, perfectly to scale.
Wow.
He had to examine the precise arrangement of cephalic setae, which are the sensory bristles around the head. He had to look at the shape of the amphids, those are the chemosensory organs on the side of the head, and the exact structure of the buccal cavity, basically the mouth.
So to look at this organism, trace it by hand, and determine that its morphological features were so fundamentally distinct from every other known nematode on Earth that it required the creation of a brand new monotypic genus.
Yes.
That requires an encyclopedic visual memory of thousands of other species.
It really does. It requires decades of expertise.
Which really adds a beautiful layer of depth to the specific name he chose for the species, Ingenia mirabilis. The Latin word mirabilis translates to miraculous or wonderful or amazing.
It does.
Applying the word miraculous to a microscopic translucent worm living in the tiny spaces of a Brazilian beach. It says a lot about the perspective of the taxonomist, don't you think?
It reflects the profound appreciation that taxonomists have for evolutionary engineering. When you spend thousands of hours staring through a microscope at the meiobenthos, you stop seeing these organisms as simple or primitive.
You see the complexity.
Exactly. You see the astonishing complexity of their biomechanics, the way their longitudinal muscles interact with the hydrostatic pressure of their pseudocellum to produce that characteristic whip-like sinusoidal movement through the sand. To an expert like Gerlach, discovering a novel arrangement of these biological systems is a genuinely miraculous event.
Let's examine the specific ecology of this organism, because the source text explicitly highlights its lifestyle, noting that the family it belongs to, Tripyloididae, is mostly free living. And nematodes, as a phylum, they often carry a really heavy cultural stigma for us humans.
Because of the parasites?
Right. The species that interact most directly with us, hookworms, pinworms, heartworms, and our pets, they're all parasitic. So we tend to think of roundworms as these terrible things.
The bias in helminthology has historically been heavily skewed toward parasitology for very obvious medical and agricultural reasons. The funding, the grants, the research hours, they naturally flow toward organisms that cause disease in humans, livestock, and our crops.
Which makes sense.
Of course. However, from an ecological and evolutionary standpoint, parasitism is just a fraction of the nematode story. The overwhelming majority of nematode species, and certainly the vast majority of nematode biomass on the planet, are free-living organisms.
So they're just out there living their lives.
Exactly. They inhabit the soil, the freshwater sediments, and the marine meobenthos, operating completely independently of any host organism.
And being a free-living marine nematode has to require a completely different suite of adaptations compared to a parasite living comfortably inside a warm intestine.
Night and day. A free-living organism like Ingenia mirabilis is fully exposed to the selective pressures of its environment. A parasite lives in a relatively stable, incredibly nutrient-rich environment provided by its host.
Free food, basically.
Right. But a free-living nematode must actively navigate that three-dimensional maze of the sand. It has to actively evade predation from microscopic crustaceans like copepods or predatory tardigrades, and it must actively hunt or forage for its own complex nutritional requirements, all while constantly adjusting to the physical and chemical fluctuations of the tidal zone we talked about earlier.
The stub actually provides specific details on that foraging behavior. It notes that they feed on diatoms and other algae. Now, diatoms are single-celled algae, but their biology is highly unique, specifically their cellular structure.
Diatoms are fascinating. They are defined by their frustules. A frustule is essentially an intricate cell wall made entirely of polymerized silica.
Silica, like glass.
They literally live inside microscopic glass boxes, and they're incredibly abundant in marine environments. They form these thick biofilms on the surface of sand grains, and they contribute a massive percentage of the Earth's primary production through photosynthesis.
Okay, but grazing on microscopic glass houses requires some highly specific biomechanical tools. How does a nematode belonging to the Tripyloididae family actually consume an organism encased in solid silica?
The feeding apparatus of diatom-consuming nematodes is highly specialized. Now, while we don't have specific morphological diagrams of Ingenia mirabilis right in front of us, nematodes in these types of foraging niches typically possess heavily cuticularized buccal cavities.
So armored mouths.
Yes. Some have complex arrangements of teeth or mandibles that are capable of physically cracking the silica frustule to access the lipid-rich cytoplasm inside.
Oh.
Others use a specialized needle-like structure called a stylet. They use it to pierce the frustule and then essentially vacuum out the cellular contents using the powerful muscular pumping action of their esophagus.
That is incredible. So this places Ingenia mirabilis at a very specific trophic level within the coastal ecosystem. It is essentially acting as a primary consumer, like a microscopic grazing animal moving through the interstitial spaces of the sand to hunt algae.
The ecological role of these free-living grazing nematodes cannot be overstated. By consuming diatoms, they are regulating the growth of the microphytobenthos. They prevent the explosive overgrowth of algae that could easily clog the interstitial spaces and completely alter the oxygen dynamics of the sediment.
They're the maintenance crew of the beach.
They really are. Furthermore, they are a critical conduit for carbon cycling. The diatoms fix carbon through photosynthesis. The nematodes consume the diatoms, converting that energy into animal biomass. And then the nematodes are themselves consumed by larger meiofauna or juvenile macrofauna.
Pushing the energy up the chain.
Exactly. They form the foundational metabolic bridge that transfers solar energy up the marine food web.
Now, understanding that ecological role requires placing the organism precisely within the architecture of biological classification. And the info box in the source material provides the full taxonomic hierarchy for us. It classifies Ingenia, starting from kingdom Animalia, down through phylum Nematode, order Areolamida, and family Tripyloididae.
And that progression down the taxonomic hierarchy represents increasing levels of morphological and evolutionary specificity.
Right. Zooming in.
Kingdom Animalia and phylum Nematode establish the foundational body plan. So we're talking about a multicellular, heterotrophic organism with bilateral symmetry, an unsegmented cylindrical body, and a complex cuticle that molts.
Basically the definition of a roundworm.
Yes. But as we move down to the order Areolamida and the family Tripyloididae, the classifications start to rely on much more minute details. Tripyloididae, for instance, are generally characterized by very specific arrangements of their cephalic sensilla.
Those head bristles again.
Right, usually arranged in multiple distinct circles around the head. And the specific tripartite, or a three-part structure of their stoma, their mouth opening. These are the incredibly subtle morphological signatures that taxonomists like Gerlach used to group these organisms together.
And they were operating on the assumption that shared morphological traits indicate a shared evolutionary ancestor?
Which was the standard practice.
But that assumption brings us to a massive, glaring structural contradiction within the text of this Wikipedia stub itself. The info box clearly lists the taxonomic rank directly below phylum as Chromadoria.
Okay.
However, the actual body text of the article states, and I quote, "This Anoplea nematode-related article is a stub." Furthermore, the metadata categories at the very bottom of the page list Anoplea genera and Anoplea stubs. So we have a direct conflict right there on the page. Chromadoria versus Anoplea.
This contradiction is honestly the most analytically rich piece of data in the entire source text.
Really? A typo.
It's not just a typo. It is a visible fracture line left behind by a fundamental paradigm shift in biological science. To understand why an organism would be simultaneously classified as Chromadoria and Anoplea, we have to look at the fourth reference cited in the article.
Oh, the 2010 paper.
Yes. A paper by H.M. Bick et al., published in the journal BMC Evolutionary Biology. It's titled Moving Towards a Complete Molecular Framework of the Nematode, a focus on the Anoplida and early branching clades.
The operative phrase in that title being molecular framework.
Exactly. For the entirety of taxonomic history leading up to the late 20th century, classification was based entirely on the kind of visual morphology Gerlach practiced back in 1957. What does it look like?
Right.
But the advent of affordable, high-throughput DNA sequencing completely shattered those morphological family trees. Molecular phylogenetics introduced the ability to classify organisms not by what they look like, but by reading their actual genetic code.
Looking at the DNA itself.
Right, often using specific markers like the small subunit ribosomal RNA gene. And what researchers like Beek and her colleagues discovered when they began sequencing the genomes of nematodes was that morphology is often profoundly deceptive.
Deceptive how?
Particularly in the Meiobenthos, the interstitial environment imposes such extreme physical constraints on the organisms living within it that completely unrelated lineages will independently evolve the exact same morphological solution.
Convergent evolution.
Precisely. Two completely different nematodes might evolve the exact same buccal, cavity, and sensory bristle arrangement simply because they both have to navigate quartz sand grains and eat silica-encased diatoms.
So visually, under the microscope, they look like close cousins.
But genetically, they diverged hundreds of millions of years ago. The traditional morphology-based classification divided the phylum Nematode into two main classes, the Adunophoria, which contained the subclass Anoplea, and the Cesarneti.
Okay.
But when molecular data was applied, that entire older system collapsed. The Cesarneti were found to be just a highly derived branch nested within a much larger clade. That whole group was subsequently reorganized into the class Chromadoria.
Got it.
And the remaining early branching, largely marine lineages, were reorganized into the class Anoplea. The 2010 Bick paper was a critical part of the ongoing effort to untangle the deep evolutionary roots of the Anoplea, mapping out these early branching marine clades using genetic sequence data.
So the contradiction on the Wikipedia page listing Chromadoria in the formal info box but leaving Anoplea in the body text is essentially a digital fossil of this transitionary period in science.
It is a perfect artifact of decentralized open-source database management trying to keep pace with a rapidly evolving scientific consensus.
Someone probably updated the taxonomical info box with a newer classification, maybe based on a massive automated molecular database bot.
Almost certainly an automated update, yes.
But the manual text in the body of the article and the associated metadata categories at the bottom remained untouched, reflecting an older or perhaps conflicting taxonomic interpretation. The disagreement between Chromadoria and Anoplea sitting right there on this single page encapsulates the ongoing, fiercely debated process of rewriting the evolutionary history of an entire phylum based on genomic data.
It's science happening in real time, visible through the version history of a web page.
It perfectly illustrates the nature of the platform hosting the information, too. The text itself explicitly acknowledges its own incompleteness. It literally states, "This article is a stub." You can help Wikipedia by adding missing information.
A stub about a monotypic genus serves as a very stark reminder of what biologists call the taxonomic impediment.
The taxonomic impediment, meaning the gap in our knowledge.
The massive gap between the estimated biodiversity of the planet and our actual catalog knowledge. Nematologists frequently discuss the nematode diversity deficit.
How big of a deficit are we talking?
Well, we have formally described somewhere around 25,000 to 30,000 species of nematodes total. But extrapolations based on deep sea and marine sediment sampling suggest the actual number of extant species could range from 1 million to over 10 million.
10 million,
Yeah.
and we only know 30,000.
Exactly. The vast majority of the biological machinery operating on this planet is completely undocumented. An entry like this, openly flagging itself as a stub, represents the actual frontier of human knowledge.
It's a placeholder.
It is a placeholder for the thousands of hours of molecular sequencing, ecological observation, and morphological description that have not yet been funded, let alone performed.
Yet despite being a stub, the global reach of this highly specific information is extensive. If you look at the sidebar of the page, it lists the other languages in which the specific article exists. It is available in Persian, Japanese, Malayalam, Turkish, and Vietnamese.
The presence of those specific languages highlights the decentralized, non-hierarchical nature of modern knowledge distribution. It's amazing.
It really is. A morphological description published in a Berlin Zoological Journal in 1957, based on a scoop of sand collected in Brazil, is now localized for readers in Kerala, Tehran, and Ho Chi Minh City.
And there is no central authority dictating that the taxonomy of a Brazilian marine worm must be translated into Malayalam. It is purely the result of thousands of autonomous, geographically distributed volunteers and probably translation algorithms working to ensure that the sum of human knowledge, however niche, is universally accessible.
The infrastructure supporting that massive accessibility is visible at the very bottom of the page, in the taxon identifiers box. This section links our worm, Ingenia mirabilis, to a multitude of global taxonomy registries.
Let's hear them.
Okay, we've got Wikidata, Wikispecies, the Catalog of Life, the Encyclopedia of Life, the Global Biodiversity Information Facility, the Interim Register of Marine and Non-Marine Genera, the Integrated Taxonomic Information System, the Open Tree of Life, the Paleobiology Database, and the World Register of Marine Species.
Those identifiers represent the semantic web in action. In the modern era, a biological organism requires a digital ontology to exist scientifically.
So Ingenia mirabilis is assigned a unique alphanumeric identifier on Wikidata, Q1571286 here.
Yes, and that identifier functions as a universal anchor point. Regardless of the language being spoken or whether the user is a human researcher or an automated data scraping algorithm, Q1571286 here unequivocally refers to the specific organism described by S. A. Gerlach in 1957.
And these interconnected databases are exactly how the naming collision with the dinosaur Yanshini is policed and enforced today, right?
Exactly. The Paleobiology Database and the World Register of Marine Species are constantly automatically cross-referencing these alphanumeric identifiers against the rules of the ICZN to flag taxonomic homonyms.
So the organism has been abstracted into a permanent data node, while the physical worm is microscopic, restricted to the tiny interstitial spaces of the Brazilian coastline, subject to predation and the harsh realities of the mediobenthic environment. Its digital avatar is immortalized within the interconnected architecture of global scientific registries.
It's a really beautiful way to think about it.
We set out to extract every piece of insight from a really brief encyclopedia entry, and the resulting journey has been surprisingly expansive.
It covered a lot of ground.
From the fluid dynamics and chemical gradients of the Brazilian coastal sands in 1957, we analyzed the incredibly demanding morphological work of mid-century taxonomy. We examined the specialized biomechanics required for a microscopic, free-living organism to hunt and consume silica-encased diatoms, maintaining the foundational balance of the marine food web.
We observed how the strict bureaucratic framework of the ICZN forced a paleontological renaming, allowing a tiny nematode to maintain naming priority over a dinosaur.
A huge win. And we navigated the profound scientific friction visible on the page itself, the contradiction between Chromadoria and Inopula, which serves as a direct reflection of the ongoing molecular-driven revolution in evolutionary biology.
The density of information compressed into those few paragraphs is a testament to the interconnected nature of scientific disciplines. You simply cannot fully understand a single sentence about marine taxonomy without eventually touching upon oceanography, molecular genetics, evolutionary history, and the digital architecture of modern databases.
So the next time you walk along a beach and look down at the sand, consider the sheer scale of what is occurring beneath the surface. Within the microscopic spaces between those grains, there are vast, complex ecosystems operating entirely outside of human awareness. Billions of organisms are navigating chemical gradients, hunting, reproducing, and driving the foundational carbon cycles of the planet.
It really forces a reassessment of what we consider significant. Because if a handful of sentences about a single monotypic genus like Ingenia can unravel the complexities of the taxonomic impediment, highlight the ongoing revolution in genomic phylogenetics, and intersect with international paleontology, what other massive paradigm shifts, what other miraculous biological mechanisms are currently hiding in plain sight, just sitting quietly as ignored stubs in the deepest archives of human knowledge, waiting for the right questions to be asked?
Keep exploring the hidden worlds.